Good morning, everyone. Thank you for joining this webinar. I'm Liz, your host for today, and I'm an Associate Director at Integrated Marketing at PacBio. Our two guest speakers today are Patrick Truong, a postdoc at the Department of Gene Technology, KTH Royal Institute of Technology, and SciLife Lab, Sweden. We also have Lieke Michielsen, postdoc at the Brain and Mind Research Institute at Weill Cornell. Before we get started, here are some housekeeping items. You will hear two recorded presentations. First, I'm going to do a very short introduction, after which you hear two recorded presentations, followed by a live Q&A. All attendees are muted. At any point in time during this webinar, use the Q&A to ask questions. To look for the questions box, look on the upper right of your GoToWebinar. There should be a little icon with the question mark, and that's where you can submit your questions. We will answer them at the end at the live Q&A portion. Also, I encourage you to check out the collateral, which is the material section, also on the upper right corner next to the questions. It's the one with a little document sign. Finally, at the end of this webinar, you will be presented with a survey. We would really appreciate if you can fill it out. Now, I'd like to first spend just 2 minutes quickly going over HiFi sequencing. HiFi sequencing is capable of sequencing large DNA fragments of up to 25 kilobases with 99.9% accuracy. It can sequence through highly repetitive regions with no sequencing bias. And if sequencing native DNA molecules, such as with whole genome sequencing or unamplified DNA fragments, you can retain the methylation signals. The unique features of HiFi enables a broad range of applications, from telomere to telomere phase genome assemblies to metagenomes. For today, we will be focusing on RNA sequencing, where alternative splicing of genes create many different transcript isoforms that are difficult to tell apart using other technology. Since most RNA transcripts have a range of average of 1 to 2 kilobase in length, this can be easily sequenced with HiFi. Finally, another relevant method for today's talk in RNA sequencing is Kinnex. Kinnex is a PacBio library preparation method that concatenates smaller amplicons, such as cDNA, from bulk or single-cell transcriptome into longer DNA fragments that can be sequenced with HiFi. This enables effective throughput increase without needing more sequencing runs. The table here below shows the recommended RNA Kinnex kit based on the average amplicon size and the throughput on different instruments. And now we will get into our two guest speakers' presentations.
Hello, my name is Patrick, and I'm a postdoc in computational methods. Just a brief background about me. I started my research career in computational methods in mass spec proteomics. And then I moved on to methods development in phylogenetic trees, and now I'm working on spatial multiomics methods. And today I'm going to present to you guys about how we're using the new spatial VDJ with Visium HD 3' and some analysis I performed on lymph node and tonsils. So before we start this presentation, I just want to acknowledge Stephanie Kravitz, Eric Leonard, Joey Arthur from 10X, and of course Elizabeth Zeng from Pacific Biosciences for providing the data and or for providing samples and providing scripts for helping me the data processing. I'm from Joachim Lundeberg's lab, and basically what we focus on is spatial omics. And we develop tools and applications and do various biological applications with it. So basically, spatial VDJ, it's an enrichment of spatial transcriptomics. In spatial transcriptomics, what we basically do is that we capture transcriptomes at different spatial locations. And in spatial VDJ, we basically, besides having the transcriptome, we take out specifically the BCRs and TCRs, and then we enrich that. These are typically sparse signals that needs to be enriched for us to be able to see what is actually going on in our immune cells. And what we're actually enriching from these transcripts are the so-called VDJ sequences, which maps the immune clones in our tissue. And then we try to link those immune clones to whatever we have in the H&E and the transcripts we see. So basically, what we're looking at is the BCR and TCR signals. And BCR signals are a signal from the B cells and the secreted antibodies that these B cells produce. And actually, the BCR is the majority of the signal that we capture with this technology. Yeah, but we also capture TCR signals, which are from the signals from the T cells, and these are cells that recognizes antigen presented by other cells. Here's a schematic of how the VDJ segments looks, or actually a BCR antibody looks. We have the VDJ segments, which codes how strong or what kind of epitope the antigen is grabbing and how strongly it grabs it. And then we have a constant region which defines the role of this specific B cell. And then in this segment, the VDJ segment, this is what something that's called somatic hypermutation happens. And that is when mutation happens in the VDJ segments and kind of to help the antigen recognize the epitope better or more strongly, so to say. And to be clear of the nomenclature used in this field, we have a clonotype is basically the initial event that creates this specific lineage of a B cell or a T cell. And then when we're trying to algorithmically define this, we use something called clone calling. And basically what we do is we're trying to group the sequences, the VDJ sequences we have, to a germline database and infer the original VDJ recombination events and then build evolutions from that. And a typical definition of a clonotype by an algorithmic tool, a very common algorithmic tool called IgDiscover, is that we have the same V and J assignments. We have identical length CDR3 sequences, and these CDR3 sequences should be highly similar. Now, relating to these immune cells, why is it so important? Well, it has been shown in a lot of studies that spatial information is actually very important for us to understand what is going on in our adaptive immune system. It has been shown in study that, for example, how B cells or the spatial location kind of decides how where B cells mature and proliferate and where they kind of do this mutation to get better antigen recognition. It's shown that how the spatial organization of these immune cells are in lymph node can help us prevent pathogenic spread. It has also shown to be important in autoimmune infectious diseases. And one of the hot things I guess is it's shown that it's very important in cancer and in how patients respond to immunotherapy. So when we're talking about cancer, we're actually talking about tumor phenotypes, and these phenotypes, they're kind of spatially dependent. And depending on how this tumor look, it can influence a lot of how the prognosis of different treatments are. What I mean with that is basically, is the tumor like immune infiltrated? Is it cold or is it inflamed? All this kind of stuff is kind of important to understand what is going on with the immune system. How does it react to the cancer? How does it react to potential treatments? And this is exactly what spatial VDJ can help us with. It can help us understand how does our immune system react to these different kinds of tumor, for example. And here are some applications of spatial VDJ. One of the things we're doing in our lab is looking at B cell clonal evolution and how that kind of relates to cancer evolution. You can kind of track how B cell might or might not follow how the cancer evolves. You can also track how B cells move, how their evolution kind of depends on the spatial context, so to say. Another thing we can do is that we can relate it to different gene programs or gene expressions to try to describe the environment around these specific clones. And if we have more data, for example, MSI data, we could relate these clones to metabolites, lipids, or peptides. And a third thing we're doing is we can kind of pair these components that we're finding, the lights and heavy chains, and try to see where can we potentially capture functional components, like complete functional components of the BCR receptors, and what is it actually binding to. And these are two of the samples that was so kindly provided to us by 10x, one of a human lymph node and one of a human tonsil. And if you look at this section, we can kind of see that there is some kind of blob structure going on. And if we relate that to the human protein atlas, we see that these blobs, they're actually germinal centers. So if we put this side by side, on the upper here, the 10x samples, and in the lower, the human protein atlas, we can see that, in fact, we have germinal centers and we have lymphoid follicles in the sections we got. So to analyze this, this is the analysis pipeline. Not going to go through this in too much detail, but in broad terms, we're pre-processing the raw data, running clone calling with IgDiscover, and then in the downstream analysis, we do some general statistics, we do light heavy chain pairing, and we do phylogenetic tree inference on the sequences we managed to get. And this is one of the results we get when we try to screen the different immune genes that we have from the data. We can see that we get this beautiful pattern across the tonsil and lymph node section. So we are basically plotting the genes related to B cells and T cell activities and then iterating that. And we can see that there's a lot of things going on. Now, if we pause it and look at a snapshot of it, we're looking at the HMGB2, which is a proliferation marker, CR2, which is a B cell marker, and CXL13, which is a follicle chemokine marker. We kind of see that if we look at this specific germinal center here, we see that the B cells are proliferating in one side of the germinal center, and it kind of moves upwards towards this follicle chemokine, which is calling the B cell. And this kind of unprecedented resolution of this technology is like a really cool feature that I don't think we have seen before with the lower resolution Visium. Now, comparing the Visium HD here to the left to the Visium 55 micrometer to the right here, we can really see that what I was talking about, that you really see the details much better when you're iterating through the genes on the high resolution one compared to the 55 micrometer resolution ones. Going into some details about the count statistics that we captured. One thing we need to note here before we start talking about numbers here is that the 55 micrometer sample here we have is actually breast cancer, while the other two are healthy samples. So we try to not look at the actual clone counts because that kind of depends on how your immune system reacts. Right, but what's interesting here is to see that in the 55 micrometer, the relative ratios of the BCR to the heavy chain to the light chain are relatively at the similar level, while the heavy chains are a lot less in the 2 micrometer. And that kind of shows that there is still some potential improvements we can do to this Visium HD in terms of keeping the longer transcripts in the Visium HD. So this kind of says that as we're going down in resolution size, we kind of lose some of the longer transcripts that we have, or we might not be able to capture them fully. If we look in terms of what are we actually capturing, we have these plots. It basically shows that in the top 20% of the clones, we're capturing for the heavy chains almost 80% of all the counts we have, and for the light chains, all around 95%, 95, 97% of all the counts. So basically, when we're looking at the footprints of the BCR, most of the stuff we see is from the 20% biggest clones, and the rest could be singletons or lowly, what do you call, lowly spread clones that just exist in the environment that might not spread themselves. We see the same patterns in the T cells, but not to the same degree. We see that most of it is captured by the same 20% top clones, but not 80, 90%, but rather like 30 to 50% of the counts is from these top 20 clones. Now, if we combine all the VDJ chains we have and plot them on a tissue section, it looks like this on the different resolution. We see that as we aggregate them, aggregate the resolution bins, we kind of get this very, very pretty pattern of that follows the morphology. Now, if we look at both these tissue sections and relate it to the actual image that we had before, we can see in the lymph node that we have these blobs, which represents the germinal centers. So in the lymph node, we see just stuff, or basically see the clonal expansion happening inside the lymph node, while in the tonsil, we see that they are kind of around the blob structures, basically showing that, or I would bet that if I would plot the not the isotypes, but the type, this would be secreted or antibodies rather than cells, and this would be a higher percentage membrane-bound B cells. And if we look at these T cells, we see the same pattern, but since we're capturing so much less of the T cells, it's a bit more diffuse. And if we compare it to the 55 micrometer, we see that while the 55 micrometer still captures the pattern that we see in the H&E image, we see that there are some blobs here on the heavy and light chain. One thing we note is that we don't actually manage to capture these kind of ridges so accurately. And the reason for this is that when we have these 55 micrometer spots, we kind of get these mixtures of different cells, and these mixtures kind of it's very hard for the 55 micrometer to accurately capture these small, fine differences. But overall, it's still pretty consistent with the H&E. Now, moving on to looking at individual clones. So here, basically, we have plotted up the top 50 clones and iterated through those. We see that in the right, we have clones that are very much confined inside germinal centers, while in the tonsils, we have a lot of clones that are kind of diffused outside germinal centers. And these could be seen as basically diffused antibodies. Now, if we look into one of those clones that are confined in a germinal center and look at this Phylogenetic tree, we kind of see that the Phylogenetic tree is rather flat. And the interesting stuff is when we kind of compare it to a clone that is in two different germinal centers. In this case, we can see that if the sequence data alone tells us that a specific clone should branch off to two different subclones, these subclones they tend to evolve in two separate germinal centers. And this kind of suggests that we have some kind of localized diversification of B cells. Now, with Visium HD, we can go even one step further. We can plot a more complex tree, like this tree I'm showing you here. We have two big sub-clades. In the one subclade, we have the red, blue, and purple. This is the upper one here. And then in the other one, we have the blue, yellow, and orange. It's not clear from the 16 micrometer, but if we scale it in and look at the 2 micrometer picture, we can kind of see how even within the subclades, we can see spatial localization of the subtrees or within the subclades inside the clades. So here we see that the red clade kind of separates to the right of this germinal center and the green one to the left. And here we see that the yellow-orange are to the top of this blob, and the blue one are more towards the bottom of it. So you get this really detailed insight into how B cells can evolve in germinal centers. Now, one other thing we have done is the heavy and light chain pairing. So it's a bit long to go into the details, but our method basically combines the sequence, the spatial VDJ, co-location, and co-abundance to infer heavy and light chain pairs. And if you plot it, we're using Bayesian method, and we get a posterior probability. And if we set the threshold at the elbow point, or the closest 0.05 to the elbow point, which is 0.85, we find 354 pairs for the tonsils and 84 pairs for the lymph node. And if we plot up these pairs, it looks like this. So here we're plotting the top 30 pairs. On the upper left, we have the heavy chain. On the upper right, we have the light chain. And on the bottom left, we have the intensities combined. And on the bottom right, this is the interesting one, where we have the x's, which marks where heavy and light, they are intersecting at the exact same pixel. And we see that every time, or at least every time of these top 30 pairs, we have a lot of spatial co-localization of the heavy and light chain. And the same thing happens here when we're looking at the same thing for the lymph node pairing. That a lot of times we see that when the pairing method says they're overlapping, they are in fact overlapping in space. And that suggests that these are actually where pairs of functional antibodies exist. Now, if we zoom it in and look at the 2 micrometer resolution of that, of a specific pair, this is actually how it looks. A red one is a heavy chain, and the green one is a light chain. And this is how it looks if you zoom it in, which is pretty cool to see that at 2 micrometer resolution, you can see the heavy and light chain pair at almost exact spatial location. So in conclusion, what have we done? We have basically, with the tonsil and lymph node sample, find evidence of localized diversification of immune repertoire. And we have shown that the spatial VGA can give us unprecedented insights into the immune evolution, and we get near single-cell resolution of the heavy and light chain pairs. And with that said, I want to thank all the collaborative partners, and again, 10x team and Pacific Bioscience for making this possible. Thank you.
Hi, everyone. Thank you for joining this webinar, and thank you to PacBio for inviting me. So I'm Lieke Michielsen. I'm a postdoc in the Truong lab at Weill Cornell Medicine, and today I'll present our most recent project about spatial isoform sequencing at a single-cell resolution. So before diving into all the details, I'd like to acknowledge my 2 other co-first authors. So Andre, he's a postdoc in the University of Helsinki, and Corinne, who's a former grad student in our lab, and without them, this work wouldn't have been possible. So in the Tilgner lab, we're mainly interested in alternative splicing, and we primarily focus on alternative splicing in the brain. So all the results I'll show, especially at the beginning of the presentation, will focus on the brain as well, but I would like to emphasize that all the methods we develop can be applied to any species that has a good reference genome, to any tissue type you want. So at the end, I'll also show some preliminary cancer data. So to make sure that we're all on the same page, a brief introduction about alternative splicing. So when DNA is transcribed into RNA, it still consists of exons and introns. And the introns, they have to be spliced out. And that can happen in different ways. So for instance, all exons can be included, and the mRNA can be translated into protein A. But it can also happen that, for instance, exon 3 is excluded and the mRNA is translated into, will result in protein B. And these proteins can have completely different functions. So it's really important to study the isoform expression instead of just the gene expression itself. And to be able to distinguish these mRNA molecules, we need long-read sequencing. In the past, we and also other labs have shown, have already combined spatial transcriptomics with long-read sequencing, and we have shown that we can capture patterns, spatial patterns of isoform variation. And both our studies relied on the Visium technology, so they use a 55 micron resolution, and both showed that SNAP25 showed spatial variability. So for instance, here we saw, we had a sagittal section of the brain, and we saw that the 202 isoform was shown more at the top, and the 201 was expressed more at the bottom. And here in the coronal section of the brain, we saw that the 201 isoform was expressed almost everywhere in the brain, while the 202 isoform was specific to certain layers. However, even though we could show now that these isoforms showed a spatial pattern, we couldn't really pinpoint which cell type actually contributed to this, because both cell types, they lack the single cell resolution. Well, luckily, there are newer techniques now that allow for the single cell resolution. So with the Visium HD, we got a 2 micron resolution, and with the Stereo-seq, we get the 500 nanometer resolution. So what we did in this project is that we combined the Stereo-seq and Visium HD platforms with long-read sequencing to be able to study spatial isoform variation at the single cell level. So for this, we had 5 coronal mouse brain slides, of which 2 were sequenced with the Stereo-seq platform and 3 with the Visium HD platform. And they were either sequenced with nanopore or PacBio, and some were sequenced with both. And as we can see here, here's one example of the Visium HD slide, one example of a Stereo-seq slide, is that it are all coronal sections, but they come from slightly different parts of the brain, but still similar brain regions are covered. So the cortex we see in both, the hippocampus is in both slides, but some parts are also some regions are also slide specific, such as the striatum, which is only found in the Visium HD. So afterwards, we used the H&E staining for the Visium HD and the DAPI staining for the Stereo-seq to do a cell segmentation. And then we annotated cell types using RCTD. So we use cell type deconvolution. And for the Stereo-seq, we had short read gene expression data as well. So we relied on that. For the Visium HD, it was unfortunately not available. So we had to use the long read gene expression, which is a bit sparse. So it was for some cells a bit more challenging to assign a cell type. So then we couldn't use them for the cell type specific analysis. Yeah, so then a bit more information about how we actually did the long read sequencing. So instead of naively sequencing the cDNA, we did a long molecule selection and exome enrichment as well to make sure that we get as much spliced reads in our data and as much long mRNA molecules as possible. And then we sequence it either with PacBio or Oxford Nanopore, or sometimes with both. Then when we compare the statistics of our enriched cDNA, which we have sort of Stereo-seq and Visium, and for the Visium HD, we also had some unenriched cDNA, so we can nicely compare if our long molecule selection exome enrichment actually works. And we see that in the enriched cDNA, the length of the reads is longer, and we also end up with more spliced molecules. So this is really useful for downstream analysis. So then the first important step when pre-processing the data is, of course, the barcode calling. Because when we end up with a bunch of reads, we, of course, have to know what the original location in the slide was, what barcode the read contains. So for that, Andre, our collaborator, developed spl-IsoQuant 2. So spl-IsoQuant 2 does the barcode and UMI calling, while isoQuant, the original isoQuant algorithm, assigns a gene, and an isoform is possible to every read. And then this information is combined to do UMI deduplication. So you end up with a sort of count matrix with for every location which isoforms were expressed. And previously, every time a new protocol was developed, the barcode calling algorithms had to be adapted as well because the molecule structure would change. However, now spl-IsoQuant is written such that it works with all the common single-cell spatial protocols, such as the Visium HD, Stereo-seq, 10x, but also slide-seq version 2. And even if your protocol of interest isn't listed, users can specify their own molecule structure now, such that spl-IsoQuant 2 can still handle it. So for instance, for the 10x kit, you can specify what the structure of a read looks like, that you start with read 1, the barcode, UMI, poly-T, cDNA, TSO, and then what these individual elements should contain. Such that, for instance, it can be a constant value, but you can also specify a wide list of barcodes, indicate that the UMI should be of length 12, but can be any combination of base pairs. And when you define this molecule structure, or if you want to develop a new wet lab protocol, you can be as creative as you want. So for instance, for the Visium HD, there are 2 barcodes behind one another, but there can also be a barcode at the beginning, at the end of the read. Everything is possible. So we applied spl-IsoQuant then to our long read data. And here I'll mainly show the results of the Visium HD data, because it's really interesting to compare our sample V1, for which we did enrich the cDNA with the long molecule selection and exome enrichment, and compare it to V2 and V3, which were naively sequenced. So for V1, we sequenced it with ONT and PacBio, and we used 1 flow cell and 1 smart cell respectively. And we started with around 100 million reads for both the nanopore and PacBio data. While for V2 and V3, it was data we received from 10x Genomics and PacBio that was naively sequenced with either 4 flow cells or 4 smart cells. So you can see that the number of sequence reads is way higher for these 2 data sets. So then the first step of spl-IsoQuant 2 is the barcode calling. So we can see that the number of barcodes, the number of reads with a barcode detected is relatively higher in the PacBio data, which is explained by the lower error rate in the PacBio data, in PacBio. Then we check how many reads can actually be assigned to a gene. Here we lose some because of multi-mapping, or maybe some reads cannot be assigned to a gene at all. Then, of course, during deduplication, we lose some reads, especially for V2 and V3, because they were sequenced probably more closer to saturation. For our analysis, we're only interested in spliced reads. Again, we see that we lose a bit more for V2 and V3 because there was no exome enrichment and long molecule selection there. And then the reads we will use for downstream analysis are the spliced reads with an isoform assigned. And we can see that even though we started with a different number of sequence reads for V1 and V2 and V3, that the number of reads for downstream analysis, the number of informative reads, is actually in the same range for all the 4 data sets. So afterwards, we use these informative reads to detect spatially variable isoforms. And for that, we rely on Moran's I. And what Moran's I intuitively does is it compares the expression of a cell, it compares it to the expression of its neighbors, and if it's the same, it will result in a higher Moran's I-score. To detect spatially variable isoforms, we rely on the relative expression. And the relative expression of 1 indicates that a cell expressed the gene and the isoform of interest, while relative expression of 0 indicates that a cell does express the gene, but another isoform is being expressed. And there can also be values in between 0 and 1, of course, if a cell has multiple reads belonging to that gene. So cells that do not express the gene are not considered during downstream analysis of that isoform. And then after calculating Moran's I for our observed data, we also do a permutation test and we permute the data 100,000 times so we can calculate a p-value and do multiple testing correction. So first I'll show you the results for the Stereo-seq data. So for the Stereo-seq data, we actually had 2 samples, sample 1 and sample 2. And sample 1 was sequenced with 2 flow cells and sample 2 with only 1 flow cell. So we see that we detect more spatially variable isoforms in sample 1, which is also as expected as the read depth was higher. And then when we combine these 2 samples, because they were consecutive slides, so we could align them, we see that that even increases the number of spatially variable isoforms. Then in the Visium HD data, we notice that when sequenced with ONT, we detect the same range of spatially variable isoforms. But for the PacBio data, the number of spatially variable isoforms increased dramatically compared to the ONT data. And this cannot be explained by the read depth because from the previous slides, we know that the read depth was pretty similar. The number of informative reads was pretty similar in these 4 data sets. Also, when looking at the percentage of spatially variable isoforms, we see that also this increases in the PacBio data. So it's not just that, for instance, more isoforms could be tested. So for V1, we know it's the same slide, but sequenced with both nanopore and PacBio. So we would expect a similar set of spatially variable isoforms. And what we see is that what we found in our PacBio data is actually a sort of superset of the 2. So our hypothesis is that the slightly higher error rate in ONT, as such, it has a slightly lower precision for the barcode calling and a slightly lower precision for the isoforms assignments, which might create a little bit of noise, which might make it more difficult to detect spatially variable isoforms. Even though we know the precision only drops from 99 to 97%, this could explain this difference. So then I'll show you some examples, because I started this presentation with SNAP25, that SNAP25 was shown before to have a spatial pattern in the brain. And in our data, we detect the same. So what you see in this plot is in gray in the background, it's the DAPI staining. In every dot, a dot is plotted if the SNAP25 gene was expressed at that location. And then the color indicates the relative expression. So a yellow dot means that SNAP25, the gene, was expressed, and that our isoform of interest, the 201 isoform, was expressed. So we see that that's mainly in these layers of the brain, in the midbrain, in the hippocampus. But the relative expression is lower in these layers of the cortex. And this means that the 202 isoform was also partially expressed there. So now we can also look at our cell type specific data, and we can see that this is actually driven by the excitatory neurons. And where SNAP25 is just a relatively easy gene with just 2 isoforms that change, we also find more complicated examples in our data. So just for instance, for the ARPP19 gene, we saw that there were 4 spatially variable isoforms. So we had the 203 and a new isoform that were specific to the cortex, while the 209 and the 205 isoform were mainly specific here to the white matter and to the midbrain. And interestingly, these 2 isoforms, they encode the same protein. They both encode ARPP16, while 209 and 205 both encode ARPP19. And what's maybe interesting to mention is that even though 205 and 209, they encode the same protein, we see from their mRNA structure that they, for instance, have a different 3' UTR length, which can influence, for instance, the mRNA degradation or the localization of the gene, and as such also affect translation. So there are still different regulatory mechanisms between these two isoforms. And then again, for ARPP19, we can also look at the cell type specific effects. So for instance, in excitatory neurons, we see that 3 isoforms are expressed. We see that 203 is mainly specific to the cortex. The new one is specific to certain layers, while 209 is specific to the hippocampus. So in excitatory neurons, we see a spatial pattern of all 3 isoforms. While for the other cell types, the inhibitory neurons, oligodendrocytes, and astrocytes, we see that those mainly express the 209 isoforms. So for ARPP19, there are cell type specific differences, and also there's a spatial pattern to be found. So these were just some examples what we found in our brain data, but as mentioned before, this can also be applied to human tissues, to disease tissues. So here are some preliminary results on cancer data. So from PacBio, we received some sequencing data from colon cancer and from breast cancer. And this data was sequenced, it was naively sequenced, so there was no long molecule selection and no exome enrichment. And in the end, we had for the colon cancer, there were 8 million reads we could use for downstream analysis, and for the breast cancer, almost 3 million reads, which resulted in 319 spatially variable isoforms in colon cancer and almost 200 for the breast cancer data set. And I'll just show you one example of what we found here. So for instance, the SQ-STM1 gene had 2 spatially variable isoforms, and SQ-STM1 encodes the P62 protein, which correlates with a worse clinical outcome. And so for this gene, there were 2 spatially variable isoforms, the 206, which does not encode a protein, but actually has a retained intron, and the 219 isoform, which actually is protein coding. So in conclusion, we show now that we can combine these single cell resolution spatial transcriptomics platforms with long read sequencing. I really recommend combining to not just naively sequence the cDNA, but to really apply the long molecule selection and exome enrichment, because it results in more useful, more informative reads for downstream analysis, and it will also reduce the cost of an experiment if you only need 1 flow cell compared to 4. There are more reads recovered in the PacBio data due to more accurate barcode calling. And with our data, we could reveal now some cell type specific spatially variable isoforms in the mouse brain. But as shown with the preliminary cancer results, we can apply this now to look for disease-associated splicing changes. So with that, I'd like to thank again Andre and Corinne for all their help for driving this project together with me. Of course, Hagen and Iman for all their supervision, and the rest of the lab for all the brainstorming and all the input. I'd like to thank you all for your attention. And if everyone is interested in collaborating on this cancer data, if is interested in especially isoforms that we found, let me know. Happy to share. Happy to answer any questions.
All right. We are gonna start doing our live QA session. The first question we have, and just a reminder, if you have any questions, please use the questions chat box on the upper right corner to ask questions. It sounds like this question would be applicable to both. How complex is the experimental workflow to get high resolution sequencing with PacBio? So my interpretation of this question, Lieke and Patrick, is just how much more work is there to get Stereo-seq or Visium HD. And Patrick, in your case, you also did TCR, BCR on top of that, not just whole transcriptome. How difficult is this to get this to work with PacBio?
So maybe I'll start answering that from a computational perspective. So I'm actually a computational person. I don't really do the wet lab procedure, so I cannot speak too much on regards to that process. But in regards to data processing, I would say it takes quite a little bit more effort. But I think you and your team at PacBio helps out a lot at making this effort smooth. It's not a standard processing with the 55 micrometer. And I guess one of the big reasons for that is the barcode structure has kind of changed from how the standard tools are, or how the standard tools recognize it. But I think this is something that you and your team are pretty on top of, or at least it feels like that from my side.
Awesome, thank you. Lieke, would you like to add anything?
Yeah, so I'm also purely computational, so I'm not sure how difficult it is to do in the wet lab. But from a computational perspective, it's now, like, for instance, for developing spl-IsoQuant 2 to do the barcode calling, that was a bit of effort since the barcodes are different in the Visium HD data, they are different in the Stereo-seq data. But now that we have this pipeline set up, it's pretty easy to apply to, if it's PacBio data or nanopore data, whatever it is.
Yeah, awesome. So I see we have a flood of questions. I'm actually thinking this possible that I will answer 2 or 3 questions that are in the same nature. So first is a question actually from me is, does PacBio support spatial applications, or is it coming in 2026? It is 2026, by the way. I think the person is still in 2025. But yes, actually, we've established official compatibility of the Kinect single-cell RNA kit with Visium HD. We are working on publishing the application note. And in fact, the data set is already publicly available, not the ones that Lieke and Patrick has presented, but it is officially compatible. Both 10x and PacBio will support Visium HD with our Kinect kits and PacBio sequencing. But then I do want to point out that for the informatics, as Patrick and Lieke has mentioned, the library prep and the sequencing is compatible, but the bioinformatics require custom pipelines. There is a Visium HD pipeline that Patrick has used that we put out on GitHub right now. And obviously Lieke has developed their own spl-IsoQuant 2. And there's a question from the attendees, Lieke, to you, is spl-IsoQuant 2 supposed to be a replacement of iso-seq preprocessing? So like, do you directly take the sequencing data into your pipeline, or do you use any part of the iso-seq preprocessing?
So what we do first is that because we use the Kinect kit, so we split the reads, and then afterwards it can be input directly into spl-IsoQuant 2. Yeah.
Awesome. So yeah, so for those who don't know Kinect, it's because we do the concatenation. We do recommend running the resegmentation, or on command lines called SCARA. It will give you the individual molecules, but then it sounds like Liike's pipeline would directly take that. And by the way, that would the segmented reads would also directly go into the GitHub pipeline that Patrick has used. So lots of options here. Let's see. Here's a question for Lieke. Are there particular, and I guess it would apply to both, but they were asking it for Lieke. Are there particular types or conditions of tissues that would yield suboptimal transcript capture? Would particular tissue preservation conditions or postmortem intervals affect data quality? I understand we're all computational, but do you have any insight into that?
Yeah, a bit. I'm not sure about the particular cell types, but I do know that, for instance, now we worked on mouse brain, so the mRNA quality is still pretty good. But if you have like post-mortem human tissues, if the mRNA is degraded already partially, you will never get, it's more difficult to get full-length isoforms, so that will lead to shorter reads and might make it more difficult to assign isoforms to reads. So for human data, or human brain data, we usually advise to not look at the isoform level, but use at the exon level, because it's more difficult to assign isoforms.
Got it.
So I agree with Lieke. I think the mRNA quality is very important for spatial VGJ enrichment as well. In fact, we recently had some samples from some samples that were processed from deteriorated, or we didn't know it was like that before. So basically, it was one sample that was used and then refrozen and used and refrozen, and for some reason, at the last step, we did expensive experiments, and that one was like when I looked into the data, you could not recover almost anything. It was like 0.001% of the counts relative to the experiments we ran before.
Got it. Here's a question, I believe, for you both. And both of you have showed in your slides, Patrick, you had those little awesome animations and pointed out the location of specific TCR clones. Lieke, you had also showed specific isoforms at different parts. How did you guys map the splice isoforms or the clones to the histology tissues that you had showed?
So actually, that's your pipeline that you provided pretty much makes the downstream analysis very similar to regular Visium 55 micrometer analysis. You have all the information there. So after processing, it should not be too hard to do it if you have worked with regular Visium before. Maybe you'll have some computational constraint because the data set is much larger than regular Visium. At least that's from my perspective.
Maybe to add a bit to that. So then we used either the Visium HD, like the Space Ranger software, or the Stereo-seq software, which maps the barcodes to the histology slide. So then you know the location of each barcode that's basically provided by the spatial platform. And then we use spl-IsoQuant too, but I guess it can be another pipeline as well, which links every read to a barcode. So for every read, you know the spatial location then. And the list of barcodes is really long because of the higher resolution for both Stereo-seq and Visium HD. So that's indeed like the memory requirements. Yeah, you need more memory and runtime to be able to run this. So.
Yeah, that would be a great time for me to continue to one of the next question is what is the minimum computational requirement in terms of CPU, RAM? Like, do you guys have any recollection of how long it took, how much memory and CPUs you used?
So from my perspective, it all depends on how efficiently you implement what you want to do and how long, what your time threshold of how slow you want your analysis to be. I actually don't have any hard number. Like my hardware has been sufficient. I haven't been thinking about it. I don't know what your perspective is, Lieke.
Yeah, so if I remember correctly, for the Stereo-seq, it was most memory intensive because the barcode whitelist is longer. There are like 500 million barcodes. We needed 128 gigabytes of RAM. But then the pipeline is pretty fast that it barcodes, or it calls barcodes in 10 million reads per hour. And then you can parallelize all of that. So I think like Minimap 2 was still the slowest step compared to the rest of the pipeline.
Yeah, I think I've heard recently Minimap 2 is surprisingly now some of the longer steps, when many years ago it was not. Here's an interesting question. How would you integrate, to both of you, how would you integrate this with proteomics data? And are there any thoughts about integrating this with proteomics data?
Ooh, what kind of proteomics data are you talking about? Like MSI peptides or MS proteomics or?
Well, the attendee did not specify, so perhaps it's just, have you thought about it?
So I've been working a bit with data integration before, or actually that's one of my current projects, MSI integration with spatial transcriptomics. Like in that sense, there are a couple of things you need to keep track of. One thing you need to keep track of is how are the different resolutions overlapping, and how are you going to make sure that they are actually measuring the same thing? You also need to keep track of how are you aligning, like that's actually a major part of it. How are you aligning the different modalities if you're working spatially? Now, if you're not working spatially, I guess it comes down to what kind of, why would you want to do this kind of multimodal integration? And then your data integration you kind of have to take the approach of what kind of problem you have to solve, I would say.
Lieke, do you want to add to that?
Yeah, so it's difficult to measure the proteomics on the same slide usually. So what you could do, I guess, is do some stainings on a different slide, but then you can only measure specific proteins or specific antibodies and maybe align that. Yeah, I think that's using another slide for it and integrate that might be easier.
So the attendee clarified that they were thinking about cytometry by time of flight. So I think that is potentially integratable, but to be honest, it's not something I'm familiar with. I have another question. We have another question let me just look at it, for Lieke. Just really quickly look. Okay, so the question was, in your talk, Lieke, you had used the word informative reads, down for the colon cancer from 200 million to 8 million. What filters did you apply to arrive at the number of informative reads?
Yeah, so it's basically the same filters as what I applied when showing all these specific bar plots. So, of course, a read needs to have a barcode, otherwise we cannot assign it to a spot. We are only interested in spliced molecules because we focus on alternative splicing. And we want an isoform assigned because we wanted to look at isoform specific analysis. So it's the same criteria as we've shown before.
Got it. Let's see. There was a question about, last question. There was a question about large fragment size selection done. I'm trying to recall, did you, because I know you did exome enrichment for some of them, Lieke, did either of you did large fragment size selection in particular on the?
Maybe it's related to the long molecule.
Yeah.
I'm not sure if that's...
Oh, I'm looking at the question, like when did you guys do large fragment size selection? Because Patrick, you just did TCR, PCR, select. Was it probe-based selection, or you actually did specific primers off that for the TCR Visium?
So I think the wet labbers did some primers to capture the TCRs and BCRs and enrich them. Like not in a selective way. I think you try to target everything.
Yeah. Okay. I think we'll answer that. It wasn't, you guys didn't do direct size selection, but you did enrich for different things, either through primers, and for Lieke, I know you had done exome enrichment for your experiments. One last question, because this was a good one. For Lieke, was there a difference in your analysis between PacBio and ONT sequencing?
Yeah, that's a good question. So for the Stereo-seq, we actually also had ONT in PacBio, and there the read depth of PacBio was a bit lower because we used the previous mass spec kit instead of the Kinnex kit. So there the ONT back then gave more results. But for the Visium HD, we used the PacBio Kinnex. And we saw more spatially variable isoforms in the PacBio data, which we think is related because the barcodes of the Visium HD data, like neighboring barcodes, are very similar. They only differ by one base pair. Mm-hmm. And that's where the lower error rate of PacBio is really important. So that's why we think there are some false negatives in the ONT data. So some missing spatially variable isoforms. But we're not 100% sure yet about what explains the difference. But the number of informative reads for analysis, for the downstream analysis, was the same, but we got more significant hits with PacBio.
Got it. Thank you both. Hope all of your attendees enjoy the webinar. The recording will be sent to you guys in a few weeks, and hope to see you guys soon. Bye.
Thank you. Bye.
Thank you.